All right, we're going to talk about chapter 14, which is biotechnology. It's a really interesting chapter, and there are a lot of applications to what's going on in the world today. I did make a lab, um, an extra lab for you guys that has some scientific articles that relate to COVID-19 and what's being done in the world today that relates to what you've learned with RNA polymerase, transcription, and then some of the biotechnology techniques we're going to talk about right now, like PCR, let's see, and different drugs that they can target to stop COVID-19. So make sure you look at that. That lab is pretty interesting, and it's going to take the place of one of the lab that we missed in our extra week of spring break. And I'll give you more information on that in Blackboard. Okay, so let's look at chapter 14's DNA technology. First thing people think of a lot when they think of DNA technology would be cloning. Cloning, you guys have all heard of the Dolly the sheep. Dolly the sheep was a completely cloned organism, an entire organism that was cloned. This is illegal in the United States, but so it can be kind of scary. But if you also think about it, identical twins, like these boys over here, they are clones. They're identical copies of DNA, cells, or organisms. So these are identical organisms or identical twins, so they are clones. And then you can think of things like gene cloning. It's not always an entire individual, an entire organism that's cloned. Gene cloning is actually very, very, very useful. There are many research purposes. There's gene therapy, a lot of diseases that can greatly benefit from gene cloning. And we're going to talk about how that works in a little bit. Okay, so how do we isolate certain genes? We don't want to, let's say we're going to do gene therapy, and we need copies of a very specific gene. We need to be able to cut that out. We cut that specific part out. So we use a tool called restriction enzymes. Restriction enzymes are able to cut the covalent bonds of DNA. Remember, the covalent bonds are on the sugar phosphate backbone of DNA. So they're able to cut those covalent bonds in very specific places so that we can target certain areas. Different restriction enzymes, they come from bacteria. Here's E. coli right here. They come from different prokaryotes and different bacteria, and they're able to cut in different spots. And they also make what we call a sticky end so that it's easy to insert another piece of DNA or some correct DNA into a certain piece. So restriction enzymes, they're like scissors. They're going to be our tool that's like scissors. They're able to cut certain areas. Okay, and we can use the pieces of DNA that we're able to cut out by restriction enzymes to make something called recombinant DNA or rDNA. Recombinant DNA contains DNA from 2 or more different sources. So what you need in order to make recombinant DNA would be a vector. The first thing is a vector. A vector is a small plasmid. Do you remember those little circles, the circular DNA that are found in prokaryotes that we've been talking about? They're little circular pieces of DNA. This is where they are important because it's pretty easy to isolate certain areas and to cut open certain areas of the small circular DNA in bacteria. They're called plasmids. And they're commonly used as vectors, which means they're like a just a delivery method. They're a way to add you can use a restriction enzyme to cut it open, add any gene, like for example right here what we're looking at, this is a human cell, so this is a eukaryote, and we're able to take a piece of a eukaryotic genome, in this case the insulin gene, and we're able to put it into this circular plasmid from the prokaryote bacterium. And we can put the insulin gene into the vector, the plasmid of a bacteria, to make something called recombinant DNA. Now, this bacteria contains the genome to make human insulin genes. Why would we do that? Well, now we can put a bunch of this recombinant bacteria in large vats and make the bacteria produce large quantities of human insulin that we can then use as medicine for people who are diabetic. So that's just one example of why we would make recombinant DNA. You do need two enzymes. So what you need for recombinant DNA, a vector, and then two enzymes. The first one is the restriction enzyme we talked about. Those are the scissors that are going to cut this gap open and also cut out this small piece of insulin gene. And then you need DNA ligase. That's going to be the glue that seals the DNA into place. So we've already been talking about restriction enzymes and DNA ligase. DNA ligase we saw in replication. Okay. Let's look at the polymerase chain reaction, or PCR. What PCR does is it takes that small piece of gene that you were able to extract using restriction enzymes. So you can take out a certain area, and you can make millions of copies of that. It's really cool because it's just this box that you can set on a countertop in a lab. And you throw all of these chemicals in. You throw the DNA sample, you throw primers, remember primase, DNA primase example. You throw nucleotides in there, A, T, C, G. Taq polymerase, that word right there is just like DNA polymerase, only it's Taq polymerase, meaning it came from a bacterium. But it's an enzyme that's going to build another copy, just like DNA polymerase. You have a buffer solution, and then you just need this little tube that it all goes in, put inside this machine. What this machine does is it heats up. So it heats up and it will separate DNA strands where the hydrogen bonds are. And then it will cool down. When it cools down, these primers, where's the primers at? Right here. The primers will go and hold it open, and they attach. And then they heat up a little bit again, not too much. And what happens is the Taq polymerase works really well in warm, hot environments, really. So they're actually found in places like the hot springs and geysers of Yellowstone, for example, in the bacterium. So this is a certain type of polymerase that works well under heat. So that's what we use it for. We heat it up again, and it goes ahead and makes another strand of DNA. So it does replication within this tiny box, which is super awesome. And it can do it, if you set it for a few hours, it can make millions of copies in just a few hours. And I have a lab for you guys to look at with this as well. It's actually in the lab, there's a video. It's really cool. So check that out. Okay, so what does PCR do? We make a whole bunch of copies of DNA. Great. What do we do with it? Well, we can use it to analyze crime scenes or analyze any type of DNA that's found. We can use it for paternity tests. We can use it for ID, any sort of crimes, in order to compare DNA to another piece of DNA. So you don't need much. You just need a tiny little bit, and you can make millions of copies. So PCR is responsible for making the millions of copies. It's not responsible for actually comparing samples to suspects. What that is something called gel electrophoresis, which we're going to talk about next. But the first step you have to do, say you get just one hair from a crime scene. Well, you're able to extract a tiny little bit of DNA from that. We need to make many, many, many copies of that DNA in order to run the test needed for a case, for example. So PCR will get you the many, many, many samples that you need. Then you can run what's called gel electrophoresis, which separates fragments of DNA by their charge and their size. So it'll produce these banding patterns. What it is, you're going to use restriction enzymes that we talked about earlier to cut those many, many, many pieces of DNA at certain spots. I'm going to show you. I think it's on this next page here. There's repetitive. Where is this? Okay, here we go, right here. Short tandem repeats or repetitive sections of DNA. They're not for, they don't code for specific traits. They used to be considered junk DNA, although we found out that there's actually many uses for those as well. They actually do perform jobs. They are important. But for now, they're not going to code for certain traits that we've been talking about. They're like repetitive segments. So it might be like, here, I think I found it right back here. No. Okay, I'll have to find a slide for you guys. But it's going to be like ATT, 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 ATT several times throughout a certain section of the DNA. And then someone else might have 20 copies of that repetitive section, where someone else might have 50 copies of that repetitive section. And if you take enough of those, every single person is completely unique. So it's a great way to compare different genomes to make sure that whether DNA matches with a suspect, for example. So what we're seeing over here on the banding patterns are the different sizes of those repetitive sections. So the farther that they travel, the smaller it is because it's harder to get through this gel. And the shorter that it travels, the larger it is, the larger the section. I'm going to show you some more examples here in a minute. Okay, so this is a gel that I actually ran in lab 2 semesters ago out in Litchfield. You can recognize your lab there. This is gel electrophoresis, and you can see these banding patterns right here. This is showing all 3 samples in one, so we have different sections here. And this is showing the different, which ones match. So you can see these 2 match right here. And these 2 match right here. And then these 2 match right here. And they're all different sizes. They've traveled different lengths throughout this gel electrophoresis. Okay, so it's a good way to separate pieces of DNA and compare them as well. Okay, another use, let's see here, biotechnology products. What can we do with this biotechnology? One thing we can do is produce genetically engineered organisms, also called GMOs. Now, GMOs are different, well, in a way, from transgenic organisms. So organisms that have had a foreign gene inserted into them are transgenic. But GMOs are just any change in the genome of an organism. So GMOs are produced from breeding. For example, the corn that you see now, it used to be teeny, teeny, tiny. I have a slide here later for that. But it was selectively bred to be large ears of corn now. That selective breeding is GMO. It's genetically engineered organism. So when you hear the term GMO, don't immediately think of transgenic. That's actually only a small section of GMOs. Now, transgenic organisms, they have had a foreign gene inserted into them. For example, Roundup Ready soybeans, things like that. Okay. Transgenic organisms are not all frankenfood like you've been hearing on the news. So some transgenic products are very, very, very useful and important. For example, we've already talked about insulin. Insulin is a transgenic bacteria where we're able to grow human insulin in these vats in order to help treat people with diabetes. More uses of transgenic bacteria. We can, what you're seeing over here, we have an oil spill, and they've actually added a gene into certain bacteria that will absorb the oil to clean up oil spills in the ocean. So that's pretty cool. This right here, they've taken an ocean poutfish that's able to live in Arctic waters, and they've put this antifreeze protein that keeps its cells healthy in the cold waters into strawberries so it can survive frost. That's pretty amazing as well.